A 22-year-old male loses his way while hiking in the woods. He has a water bottle, but no food prepared. 2 days later, he finds his way out and makes his way to a clinic for emergency care. At this time, which of the following enzymes is most likely converted to an active form? Choice A, hexokinase. Choice B, glucokinase. Choice C, phosphofructokinase 1. Choice D, phosphofructokinase 2. Choice E, pyruvate dehydrogenase. Choice F, pyruvate carboxylase. Choice G, glycogen synthase. Choice H, acetyl-CoA carboxylase. I'll wait 5 seconds and then answer the question. Alright, so here we got everyone's favorite topic. It's biochemistry. So it's not hard for you to identify the general thrust of this question. This patient's in the fasting state. So really, all we gotta do is go down the list of answers and figure out which ones are fasting and which ones are fed state. So let's just start off with just saying the correct answer. It's choice F, pyruvate carboxylase. So before I dive into each one of these answer choices, let's try to just tell the story of what happens during glycolysis. Because when it comes to questions like this, that's really the only way you can tackle them. Obviously, in addition, you also want to do tons and tons of questions until it just becomes second nature to you, essentially. But let's just run through it, okay? And we're starting with fed state. I have a bunch of glucose in my blood, and my body releases insulin to take that glucose into the cells. Glucose gets phosphorylated into glucose 6-phosphate, either by hexokinase or glucokinase, which of course is choice A and B here. Glucokinase is just basically like a special form of hexokinase in the liver. And this is just an extra detail, but glucokinase has a higher K sub M and a higher V max, which is to say it has a lower affinity for glucose, but it has a higher limit of how fast it can process glucose. So that's why they say glucokinase is only active when you have a lot of glucose in the body versus hexokinase. So putting in simpler terms, it takes more glucose for glucokinase to get going, but there is a higher speed limit on glucokinase as compared to hexokinase. Glucose 6-phosphate then gets isomerized into fructose 6-phosphate. That step isn't so important, but the next step, where fructose 6-phosphate gets converted into fructose 1,6-bisphosphate, is important. This is handled by choice C, phosphofructokinase 1. This is the rate-limiting step for glycolysis. Now, choice D, phosphofructokinase 2. For those of you who have not studied very hard or you kind of forgot about it, this can be a little bit confusing. But when your body has a ton of glucose, and insulin, which ends up leading to a bunch of fructose 6-phosphate downstream, you not only convert it to fructose 1,6-bisphosphate using PFK1, but you also convert it into fructose 2,6-bisphosphate using PFK2. Now, the weird thing here is that fructose 2,6-bisphosphate is an allosteric upregulator of PFK1. So what that means is that if I have a ton of fructose 6-phosphate, PFK1 and PFK2 both get more active, and the products of PFK2 actually come back and upregulate PFK1 to make it go even faster. So it's like this self-regulatory system where the body realizes it has a lot of glucose and therefore a lot of fructose 6-phosphate coming in, and it has this self-resolving system which feeds back into itself and tells itself to go faster. Bottom line, both PFK1 and PFK2 are anabolic enzymes upregulated by insulin. Okay, so now we have a bunch of fructose 1,6-bisphosphate. Now, there's a bunch of steps after that I feel like are way lower yield. And by all means, if you want to go take a look at them, then go ahead. But I'm going to kind of skim over them here, okay? So fructose 1,6-bisphosphate goes to aldolase, and then it turns into glyceraldehyde 3-phosphate, goes to 1,3-biphosphoglycerate through glyceraldehyde 3-phosphate dehydrogenase, and then it goes to 3-phosphoglycerate through phosphoglycerate kinase, goes to 2-phosphoglycerate through phosphoglycerate mutase, and then enolase turns it into phosphoenol pyruvate. I know I went through a lot there, but like I said, those are lower yield, so don't worry about them too much right now. Point is, I jumped over a bunch of steps after fructose 1,6-bisphosphate, and now we're at phosphoenol pyruvate, also known as PEP. Okay, this one is important because the next step after PEP is pyruvate kinase. which converts PEP into pyruvate. Now, pyruvate is super high yield. It is very useful for you to know what can happen to pyruvate because pretty much all of them are high yield. So what can we do with pyruvate? Well, first, we can do the obvious, the first thing you learn, which is pyruvate dehydrogenase, which is choice E here, turns pyruvate into acetyl-CoA, which is used in the Krebs cycle. And of course, the Krebs cycle, that's its own can of worms, and I'm not going to dig into that for this video. That's probably way too much. Let's focus back on pyruvate, okay? Like I said, what else can we do with pyruvate? Well, pyruvate can turn into lactate via lactate dehydrogenase, and this is a reversible reaction. It can go both ways. And of course, with lactic acid, there's all kinds of stuff we can talk about there too. What else can pyruvate do? Well, pyruvate can turn into alanine via alanine transaminase, ALT, your liver enzyme, right? And this is also reversible. It can go both ways. So that's why when questions ask you, for example, about a gluconeogenic amino acid, a safe choice is alanine. And then finally, pyruvate can also turn into oxaloacetate via pyruvate carboxylase, which is choice F, the correct answer here. And some of you are going to be a little confused right now. So let me try to clarify here, okay? As a general rule, when you're thinking, how do I get back from a stored energy source, whether it's pyruvate or glycogen or your triglyceride storage, like, okay, how do I go back up so I can reach glucose again? A lot of times you'll think, okay, I'll just go backwards on the reaction I just had. However, that doesn't work with pyruvate because the conversion of pyruvate from PEP is irreversible. So, like I said, PEP turns into pyruvate through pyruvate kinase. That cannot go backwards. So, in order for me to go back up to PEP, I have to take pyruvate and turn it into oxaloacetate through pyruvate carboxylase, and then oxaloacetate can turn back into PEP through phosphoenolpyruvate carboxykinase. And only then I will have PEP back, and I can go back up through the same reactions I got going down to get to PEP from glucose in the first place. I know that may have been very confusing, but for those of you who are bad with your biochemistry, I encourage you to go find one of those maps to glycolysis and Krebs cycle and such, and then follow along what I'm talking about. Once you see it and you really commit it to memory, these questions actually become pretty easy. So once again, re-emphasizing the main learning point here, PEP to pyruvate is irreversible. Therefore, to get back to PEP from pyruvate, I have to first turn pyruvate into oxaloacetate via pyruvate carboxylase, and then take oxaloacetate and convert it back into PEP through PEP carboxykinase. Then it can go back up through the same method that it got to PEP in the first place to turn back into glucose via gluconeogenesis. And of course, in this question, I have a guy who was lost in the woods. He is in fasting state, and he's going to be going through all kinds of gluconeogenesis. So of course, he's going to be turning pyruvate back into oxaloacetate so I can go back up and turn it back into glucose. That's why choice F is the correct answer here. All right, let's just finish off the other answer choices here. Choice G, glycogen synthase. I mean, this is obviously wrong. Just from a basic intuitive perspective, you should understand that if I'm starving, why would I be synthesizing more glycogen? That doesn't really make sense. I would want to be doing the opposite. I want to be breaking down glycogen so I can get the energy that I'm not getting through food. And that enzyme would be glycogen phosphorylase. Choice H, acetyl-CoA carboxylase. This is the enzyme that converts acetyl-CoA to malonyl-CoA, which is used in fatty acid synthesis, which again, that's going in the opposite direction of what we want, right? We want to be breaking down fatty acids, not making more of them. The opposite direction would be hormone-sensitive lipase, which could have been a correct answer. And you should also know that when you break down fatty acids, the glycerol form is gluconeogenic, but the free fatty acids can be only converted into acetyl-CoA, which is not gluconeogenic and will be converted into ketone bodies. So much else we could talk about here, right? Just some other random facts I'm going to throw in here real quick. Glucagon is GS-coupled receptor. So if a question talks about protein kinase A or CAMP in the context of this fasting fed state kind of question, it's talking about glucagon, right? Because insulin would be a tyrosine kinase receptor. Another general rule is that glucagon generally phosphorylates enzymes, whereas insulin dephosphorylates enzymes. And this is regardless of their function, okay? So that's kind of one of those weird things where gluconeogenic enzymes become active when phosphorylated, but glycolytic enzymes become inactive when phosphorylated. And the opposite holds true for insulin dephosphorylating enzymes, right? And this is just a general rule, right? By all means, we could go out and look for exceptions to this, but for step exam, general rule is that. And one last random thing I guess I'll throw in here. Since we have choice A and B, hexo/glucokinase here, the opposite enzyme of that would be glucose-6-phosphatase, right? And that's the final step of gluconeogenesis, which forms glucose. And of course, if it's deficient, you have what's called von Geroch's disease, right? Which is glycogen storage disease type 1, autosomal recessive disease, where you get really bad hypoglycemia. Obviously, I'm not going to dive into all the glycogen storage diseases here, but I felt the need to bring that up. This video is long enough as it is. So once again, let's emphasize the main learning point here. When you get questions like this, identify whether it's a fasting or fed state, and then go through the enzymes. And if all else fails, just start eliminating ones that you know are obviously wrong. And one testing point that they do really like is just all the stuff around pyruvate and how pyruvate factors into gluconeogenesis, because it can't go back to PEP. You have to turn it back into PEP via pyruvate carboxylase into oxaloacetate, and then oxaloacetate back into PEP via PEP carboxykinase. Alright, that's all I gotta say. I hope this video was useful. I'll see you guys in the next one.